And those who are here, thank you for coming. And those who are in Orlando for the American Heart, you'll meet a very good lectures. You'll miss it. So anyway, yeah, many people are in Orlando already, because you know the American Heart is encroaching on the previous week more and more and more. It used to start on a Sunday, now it starts on a Friday. And that's what happens. I'm really delighted to have Professor Connie Bezzina with us today, and she came here all the way from Amsterdam, where there is really a fantastic history of cardiovascular research and clinical care. I don't know how many of you know, but Dirk Doer, who really was a pioneer in treating cardiac arrhythmias and using electrocardiography in the treatment of cardiac arrhythmias, and was really the first, his laboratory was the first to map the electrical activity in detail of the human heart in 7 isolated hearts that were extracted from people who died from reasons other than heart disease. So that was really a major contribution to our field, and then it continued with many other people in the same department, and things of course are evolving as science is evolving, and now that the department in Amsterdam is really well known for making a connection between the genetics of cardiac arrhythmias and the consequences in terms of phenotypes and arrhythmogenic, what they call electrical diseases. So Connie has really been instrumental, and she heads the laboratory that actually explores the genetics of cardiac arrhythmias and trying to connect it to the electrophysiological phenotype, and she's made beautiful contributions to this whole entire area. In terms of her history, her education was in Malta, and those who know where Malta is, it's a very small island next to Sicily. Those who don't know where Sicily is, it's a little bigger island close to Italy. So that's where she's from. And then she, through England, found her way to Amsterdam, where she's been since. I just wanted to, so welcome, and we're really thrilled to have you. I just wanted to share this because this is very timely and related to her talk. This just came to my office. It's the latest issue of Science, and it's a special issue that is on mutation, I'm reading, mutation and human disease, revealing pathology with genome sequencing. So.
Thank you very much, Professor Rude, for that really kind introduction, and I'm really happy to be here in this really fantastic and prestigious place, and I've had really great discussions throughout the day. It has really been stimulating. Thank you very much. So I'll be talking about some of our recent work on the genetics of sudden cardiac death and how we're shifting our thoughts on the genetics of inherited rhythm disease from a Mendelian point of view to a more complex inheritance paradigm. I think this topic doesn't need an introduction here, but nevertheless, the overarching aim of our research is to understand the genetic predisposition of sudden cardiac death. And why is this important? Sudden cardiac death is a very important cause of mortality in the general population. Around 20% of individuals die suddenly. And in the US alone, it accounts for about 400,000 cases per year. And also, sudden cardiac death is also an important cause of cardiovascular mortality. And by far, the most important arrhythmia causing sudden cardiac death is ventricular fibrillation. Now, sudden cardiac death can strike at any age in the general population, and although it usually affects individuals in the older segment of the population, it can also strike younger individuals. Now, in the young, sudden cardiac death typically occurs in the setting of inherited cardiac pathologies that have classically been divided into the primary electrical diseases, where the arrhythmia, the ventricular fibrillation, occurs in the setting of a structurally normal heart, and the cardiomyopathies, where the arrhythmogenic substrate lies in the abnormal structure of the myocardium, such as hypertrophy. In the older segment of the general population, sudden cardiac death typically occurs in the setting of either an acute myocardial infarction or the consequences of a myocardial infarction, such as scarring and heart failure that can develop in this setting. Now, because myocardial infarction and ventricular fibrillation in the setting of myocardial infarction typically strikes at an older age, one can imagine that environmental factors over here probably have a much greater contribution to susceptibility for sudden cardiac death. On the contrary, in sudden cardiac death affecting the young, because this usually happens already in childhood and it segregates within families, we believe that there is a stronger genetic contribution to the risk for sudden cardiac death. So although we've also started doing some work on genetic predisposition to ventricular fibrillation in the setting of acute MI, most of our work focuses on sudden cardiac death in the young, because we believe that there is a stronger genetic component. We're likely to have more chance of identifying genetic factors over here, and these could eliminate mechanisms that then we could use to understand more predisposition for sudden cardiac death in the older segment of the general population, which is more difficult to dissect. So, in the last 20 years, people working in the field have provided a large body of knowledge concerning the genetic underpinnings of sudden cardiac death in the young. And essentially, we have learned that we can identify mutations that cause these disorders, and these mutations are typically very rare in the general population and carry a large effect. So essentially, if you identify such a mutation and a program within a family, individuals that carry the disease are expected to have the familial mutation. And this work has basically taught us that the primary electrical diseases are largely disorders of ion channels, they're also called ion channelopathies. The cardiomyopathies are typically caused by mutations in the contractile apparatus of the cell, and arrhythmogenic cardiomyopathy, also called arrhythmogenic right ventricular cardiomyopathy, is a disorder of the desmosome where components of the desmosomal complex are mutated. And all this knowledge has culminated in the last years by statements by the Heart Rhythm Society and the European Heart Rhythm Association on how genetic testing can be applied in the treatment of these disorders. However, in spite of this large body of knowledge that has become available for these disorders, there are some very quite outstanding challenges. One of these challenges is that we observe a large variability in disease manifestations among mutation carriers. And this is an enormous obstacle in patient care concerning risk stratification of patients for sudden cardiac death. Now, of course, one can imagine that if you have two patients with long QT syndrome and they differ in their phenotype, in the severity of the phenotype, one could argue that this could be because one patient has a gene mutation in gene A and the other patient has a mutation in gene B. Or if they both have mutations in gene A, that one has one particular mutation and the other one maybe has a more severe mutation. So the best setting to observe the variability in disease expression is actually among mutation carriers that come from large families, and essentially all the mutation carriers within these large families will harbor the same mutation. So what I have here is data from the group of Peter Schwartz, generated in the South African founder mutation family, carrying the KCNQ1A341V mutation. And what you observe over here Here we have mutation carriers, non-carriers, and this is the extent of QT interval prolongation. And as expected, most of the mutation carriers have a QT interval that is prolonged. However, you can observe that there are some mutation carriers that are actually protected somehow and have a QT interval within the normal range. Those that have an abnormally prolonged QT interval, there is a large spread in the variability. There's a large variability in the extent of QT interval prolongation. And we also know that the longer the QTC, the more is the patient at risk. Now, if we focus on the mutation carriers alone, although all these patients have the mutation, only not all patients actually develop the arrhythmia. So essentially, this variability in disease expression forms an obstacle in patient care, because how can a clinician decide who is at risk, because essentially they all carry the same mutation. The second outstanding challenge that we face in the field is that some disorders have essentially defied genetic elucidation, despite years and years of research on the topic. And one of these disorders is the Brugada Syndrome.
So Connie, in the previous slide that you showed,
this one?
No, the previous one.
Yes.
One possibility is the QT interval is just not a good index of symptoms.
It is the best.
People can have long QT, but
yeah.
I think there may be other better biomarkers that will tell you who is at risk and who is not at risk. And the QT interval per se is maybe not a very good index.
But it is the best marker at the moment.
Non-invasive.
Yeah, non-invasive, absolutely, yeah. Okay, so Brugada syndrome is described in 1992 by the Brugada brothers. There are 3 of them. Only 2 are involved in this initial report, Pedro Brugada and Josep Brugada. What is Brugada syndrome? It presents with hallmark ST segment elevation in the right precorder leads, many times in conjunction with conduction defects such as prolonged PR interval and prolonged QRS duration. The ECG may be transient and may be evoked by sodium channel blocker challenge in latent cases. And sudden cardiac death from ventricular fibrillation in this disorder typically occurs at night. Males are at more risk, and usually ventricular fibrillation strikes around 40 years of age. The disease mechanism remains obscure and is debated. And actually there are 3 proposed mechanisms for the Brugada syndrome. This is the first mechanism that was proposed by Vancelovich and co-workers, and essentially this hypothesis revolves around the difference in action potential, morphology and duration across the wall of the ventricular myocardium. So the epicardial action potential differs from the endocardial action potential because it has a strong transient outward current, which gives it this signature notch over there. And essentially what this hypothesis holds is that if you have a net increase in repolarizing force, this would preferentially abbreviate the epicardial action potential, which leads to a voltage gradient across the ventricular wall, and the current flow between endocardium to epicardium would lead to this ST segment elevation on the ECG. And this hypothesis is based on the wedge preparation and pharmacological manipulation of these currents. The second hypothesis is the depolarization disorder hypothesis, which was proposed by the Amsterdam group and is supported by data obtained in patients with the disorder. And basically, in this hypothesis, we think that Brugada syndrome is essentially a slowing of conduction that preferentially affects the right ventricular outflow tract. So the right ventricular outflow tract gets activated late, and the current flow will cause, between the area of early activation to the area of late activation, will lead to the ST segment elevation. And that hypothesis is supported by the identification of loss of function mutations in SCN5A. And loss of function mutations in SCN5A causing Brugada syndrome are truncating mutations, stop mutations, or as missense mutations that affect the biophysics in such a way as to result in loss of function. Loss of function of the sodium channel would decrease the absolute velocity of the action potential and thereby slow conduction. The third hypothesis is that which stems from the data that has been recently reported by Dr. Rudy using the ECGI system in 25 patients with Brugada syndrome, and using the ECGI they could provide panoramic views of activation and recovery of the heart. And what you observe over here, so while this is a normal heart, these are two Brugada hearts, and what you observe is that the RVT gets activated very late. What they also found for the first time in a human Brugada syndrome heart is that the RVOT not only displays late activation, but also displays prolonged repolarization in the form of activation, of prolonged activation recovery intervals. So for the first time, this hypothesis now brings together both the depolarization and the repolarization defect. So essentially, because we have these three hypotheses, there's debate about the mechanism, we feel that gene discovery in Brugada syndrome will not only help us in improving the risk stratification of patients with the disorders, but also will provide us leads to understand the basis of the ECG in the disorder and arrhythmia susceptibility. So going back to the genetics of Brugada syndrome, I told you that this disease has defied genetic elucidation, and the only gene that is by far unequivocally linked to the disorder is SCN5A. So SCN5A encodes the alpha subunit of the cardiac sodium channel in heart, and that gene is unequivocally involved. There are a few other genes which together account for less than 5% of patients, but by far the great majority of patients with Brugada syndrome remain genetically unresolved. Now, there have been several issues that have hindered gene discovery in this disorder. For instance, linkage analysis in families with Brugada syndrome has essentially failed. So assuming a Mendelian pattern of inheritance, where we assume that everybody who's affected in the family must carry a large effect genetic defect, that approach has failed. Another factor is the fact that many cases are sporadic and you essentially cannot do any family studies because you only have one patient per family. So together, these two issues, but also the fact that we see very low penetrance and variable disease expression in families who have an SCN5A mutation, made us think that maybe this disorder is not Mendelian after all. And this is an example of one of our families with Brugada syndrome that carries a mutation in SCN5A. So this is the mutation. Everybody in this family with a plus carries the familial mutation. And what you see over here, that while this individual has Brugada syndrome, another individual with the same mutation in the family has conduction disease, whereas others have both Brugada and conduction disease. So this is the variable disease expression. However, we also have this individual over here who has the familial mutation and no Brugada syndrome. And then we have this case, which is even worse, who has Brugada syndrome, conduction disease, and not the familial mutation. So this is what we've been struggling with. And this is why we started to think, okay, maybe the Mendelian model is not the right model for this disorder. So the question we asked, do some of the inherited rhythm disorders, so far assumed Mendelian, have in fact a more complex inheritance? So this is the way we're thinking now of these disorders, of the likely genetic architecture. So this is the classical Mendelian model, where you have the familial mutation. You have the mutation, you should have the disease. And this is the way we're starting to look at them now, as oligogenic or even perhaps polygenic, where you need the accumulation of a number of susceptibility variants to reach the threshold to have clinical signs of the disease. Now, these variants may actually be present in the general population. They may be common, and by virtue of the fact that they are common, they're likely to have a small effect on risk, and they may be rare and have maybe a moderate to large effect on risk. And we've started to look at the easy thing and the cheap thing to do. It's It's much more cheap to search for those than to search for these. So we started with the simplest approach and started looking for the common variants with small effect by means of a genome-wide association study in Brugada Syndrome. So this is work of Yuka Mizuzawa, who's finishing her PhD now in my group, and Julian Bark, who was with us for 4 years from Nantes. And basically, we've conducted a classical case-control genome-wide association study where the discovery stage consisted of around 300 patients with Brugada syndrome, and the controls were general population controls. Now, Brugada syndrome is a rare rhythm disease. We couldn't do this on our own, so we established an international consortium that brought together centers from different places. And at the moment, we have a database of 200 Brugada syndrome probands that are highly characterized with the same parameters in one database. And I think that this is a very important resource that will keep, I think, providing us with discoveries in this disorder. So what does a genome-wide association study do? It will compare the frequency of thousands of single nucleotide polymorphisms spread across the genome between cases and controls. And the higher than expected incidence of the SNP in the disease population will suggest that that SNP is associated with the disease. So this is the principle of GWAS, and this is the Manhattan plot that emerged from our GWAS on Brugada syndrome. For those of you not familiar with the Manhattan plot, what we have over here, each of these dots is each of those that hundreds of thousands single nucleotide polymorphisms that we analyzed. And they're spread across the autosomes and the X chromosome over here. And what you see here is the negative log of the p-value for association with Brugada syndrome. And this is the magical line over here. That is the p-value threshold of less than 5 times 10 to the minus 8, which is essentially correcting for the multiple tests, many tests, for the 1 million tests of independent haplotypes in the genome that you must correct for. We were very happy to find signals because by comparison to the enormous GWASs we see, for instance, in coronary artery disease and diabetes and many phenotypes, 300 is extremely modest. So we were very glad to identify signals, and actually we had signals on chromosome 3 and one signal on chromosome 6 over here. And what you see here basically is a zoom in on those plots. This is the data. So these are 3 loci over here, 2 on chromosome 3, 1 on chromosome 6. This is the risk allele frequency in the cases and in the controls. And these are the association p-values over here, and the odds ratio that is associated with the risk allele. What you observe here, this is one SNP that is actually not passing the 10 to the minus 8 threshold. Now this SNP we took along because we recognized it as being previously identified in GWAS on PR and QRS interval in the general population. And since we know that conduction could play a role in Brugada syndrome, we upgraded that to the replication stage of the study. The replication stage had 2 stages actually. We did the first replication in an independent set of Brugada syndrome patients of European descent, as was the discovery stage. Some of the p-values even reached genome-wide significance in the replication stage alone. And we also had the chance to replicate our findings in a set of Brugada syndrome patients from Japan. And this is the meta-analysis, and I think you agree with me that those are very convincing association p-values. We were also curious to see what the cumulative effect of the risk alleles were in aggregate. So what you see here is the odds ratio, and here is the number of risk alleles. And what you can see is that individuals that carry 5 or 6 risk alleles, so the people carrying 6 will have 2 copies of the risk allele at each locus, have an odds ratio of 20 of having Brugada syndrome, as opposed to those who carry 1 or less risk allele. So this is quite nice also, with an eye in the future on clinical applicability, although of course this is the discovery sample and this needs to be validated in the future. Now, as I said, besides gene discovery being useful for risk stratification in the long term, we're also very interested in looking in detail at these loci and looking at the genes at these loci to provide us with leads to try to understand the mechanism of the disorder. So what are the genes at this loci? So this is the chromosome 3 locus. You can immediately see over here SCN5A, which is already linked to disease. This gene carries rare variants, and neighboring SCN10A over there. Now this locus actually caused quite some interest in the field, because while the SNP in SCN5A was not unexpected, the SNP in SCN10A was quite a surprise. SCN10A is also a sodium channel gene, expressed in periphery sensible neurons and linked to nociception. And in fact, there's even another sodium channel gene over there, and these probably arose during evolution by gene duplication. So a lot of people were thinking, could it be that this SNP acts, functions, or modulates cardiac electrophysiology through SCN10A? Now, it happened that at the same time, Vincent Christophels, who's my colleague at the AMC, and Ivor Moscovich in Boston were busy looking at the regulatory landscape in the chromosomal region harboring SCN5A and SCN10A. So what they were interested in finding were actually enhancer elements along this stretch of chromosome that impacts on the level of expression of SCN5A. And they were doing this largely by two different technologies. One is ChIP-seq, and they were doing ChIP-seq for TBX5 and TBX3, which are transcription factors that are known to be very important for SCN5A expression in heart. And they were also doing this by chromatin conformation capture, which basically tells you the three-dimensional, the contacts of chromatin, of a stretch of chromosome in 3 dimensions. So basically, they identified TBX5 and TBX3 binding sites along the stretch of DNA, and they also found contact points between enhancer elements in this area and the promoter of SCN10A and the promoter of SCN5A. This happened around the same time that our GWAS was published, and the GWASs on PR and QRS were published in the general population, so they could immediately overlay the SNPs from GWAS in 5A and 10A with this epigenetic data. And what they immediately saw was that the SNP within the SCN10A locus actually resided within a TBX5 and TBX3 binding site in 10A. And you can see over here the nucleotide which is affected is this G over here. It changes to NA, and by doing so, it affects a highly conserved G nucleotide within the TBX3 and the TBX5 binding site, making it a highly likely candidate for a functional effect. And also, this enhancer element essentially makes contact with both the SCN5A and the SCN10A promoter. I will not go into this in detail, but essentially through BAC transgenesis, they demonstrated that that enhancer element is essential for SCN5A expression in heart, and they also mutated specifically the G2A change in this enhancer element, and they also showed that this was necessary for a full recapitulation of SCN5A expression in heart. So I think that this provides very strong evidence that although that SNP is located within the neighboring SCN10A gene, it acts by modulating the level of expression of SCN5A. So in this model, the G allele would give you normal SCN5A expression. The risk allele, the Brugada risk allele, would decrease the level of SCN5A transcript. Now, if that's the case, we should be able to find a correlation between the nucleotide at that locus and the level of expression in heart of SCN5A. This is data that we generated in about 100 normal, 100 donor hearts. And what you can see here is that the individuals that carry the risk allele, the A allele, homozygously or heterozygously, have less SCN5A transcript in their heart. Now you would say, but you told us that that enhancer element not only binds 5A, also binds 10A. Why doesn't it then also impact on 10A transcript? It might in other cells, but we don't think it's doing this in the cardiomyocyte because essentially the SCN5A transcript in heart is at least 1,000-fold lower than that in SCN5A. So if it's impacting on the level of SCN10A transcript, it probably has no functional impact, at least we think in the cardiomyocyte. Okay, so basically what did this locus tell us? We knew already that rare variants in SCN5A give you Brugada syndrome. What we now know that there are also common single nucleotide polymorphisms there that also contribute to Brugada syndrome susceptibility. So the rare and common variants at this locus converge on a decreased NNV1.5 function, decreased upstroke velocity, and conduction slowing. Now we go to the other locus, which is probably more interesting because this locus was never associated with arrhythmia. A gene that drew our attention immediately within this locus was the HEY2 gene. So this is a zoom in of this locus. There are other genes there that drew our attention because it was a developmental gene for which quite some data was already available in heart. It had an established role in heart development. So what was known about this gene at that time? It is a transcriptional repressor downstream of notch signaling with expression that is restricted to the ventricle, and expression of HEY2 in the ventricle decreases postnatally. HEY2 knockout mice have been published. They've been published in an inbred genetic background. Loss of HEY2 in an inbred genetic background leads to death in the first week of life. And also, these mice have a whole spectrum of congenital heart defects, septation defects, valve abnormalities. They have a very thin walled myocardium, and they have vascular abnormalities. And also, it's quite dependent on the genetic background of the mouse you're looking at. But they've also been generated, or else outcrossed, into outbred mice. In outbred mice, the phenotype is less severe, especially the developmental phenotype is less severe. They only have ventricular septal defects, and also not all mice, only about half of them will get ventricular septal defects. They survive into adulthood, and the interesting thing is that in mice without ventricular septal defects, so a structurally normal heart at birth, they go on to develop cardiomyopathy and contractile dysfunction at around 12 weeks of life, even in the absence of a VSD. And this essentially suggests that HEY2 may have a role in the maintenance of normal function in the adult heart. So this gave us the idea to ask the question, is HEY2 perhaps also involved in the regulation of cardiac electrical function in the adult heart? And this is what we set out to find out. We've done this work in HEY2 heterozygous knockout mice because we wanted to work as much as possible in a clean heart where there's no congenital heart disease and where there's no cardiomyopathy, even in adulthood. So we did these studies in HEY2 heterozygous knockout mice at 12 to 16 weeks of age, and we've used optical mapping on the lung and perfused heart, and we've also conducted cellular electrophysiology. This is optical mapping in the isolated ex vivo heart. This is work done by Carol and Bremer. And what we observed, which was of relevance for Brugada syndrome, is conduction slowing that hastened conduction that specifically affected the right ventricular outflow tract, which is of relevance to Brugada syndrome because this is the region where human mapping studies have shown us conduction slowing, and also the region where arrhythmias arise in Brugada syndrome. So this is the data over here. So if with loss of HEY2 we get enhanced conduction, we think that the allele which is predisposing to Brugada syndrome should then be a gain of function allele, because we should then get conduction slowing. We also isolated cardiomyocytes from the right ventricular outflow tract. This is patch-clamp data done by Ari Verkerk. And what we observed that loss of HEY2 prolongs the action potential duration over here. This is action potential duration at 90% repolarization. There's also some prolongation at 50% repolarization, but also in line with the improved conduction, we also see an increase in action potential upstroke velocity in the HEY2 knockout, heterozygous knockout mice. Another thing that had been reported for HEY2 is that in the ventricle, this is embryonic day 12.5, it is highly expressed in the subepicardium. So this is an embryonic heart, this is the trabecular myocardium, this is the compact subepicardium, and what you see is that HEY2 is highly expressed in the subepicardium and absent from the subendocardium. And what had been already described by several groups is that HEY2 could be involved in gene patterning across the myocardial wall. So this is NPPA, ANF, which is highly expressed in the trabecular myocardium. It is not expressed in the subepicardium. This is embryonic day 16.5. So what happens in HEY2 knockout mice, you remove the repressive effect of HEY2 from the subepicardium, and you get extension of the trabecular gene program into the subepicardium over here. So it basically flattens gradients of gene expression across the ventricular wall. Now this pattern over here is reminiscent of ion channel patterning across the ventricular wall. So we started to think, could also HEY2 impact on transmural gradients of ion channel expression in the adult heart? So this is a review from Bas Balkans, which summarizes what is known. This is the subepicardial action potential, this is the subendocardial action potential. Basically, this is shorter in duration, has a pronounced notch as compared to the endocardium. And we know that the transient outer potassium current contributes to that notch. And we also know that the SCM5A gene is more expressed in the endocardium, where it contributes to the faster upstroke in the endocardium. So we wanted to investigate this transmural gradient. We would have preferred to do this in the right ventricular outflow tract because of the relevance for Brugada syndrome, but this is impossible because the right ventricular outflow tract is very, very thin. It's impossible to dissect it and distinguish between epi and endocardium. So we did this work in the right ventricular free wall, where we distinguish between epicardial cardiomyocytes and endocardial cardiomyocytes, and we measure these by perforated patch clamp at 37 degrees. In this left panel, you see the data in wild-type mice. So this essentially recapitulated what was known from the literature. The endocardial action potential is longer, and so that is over here, longer action potential, and also it has a faster upstroke velocity. Am I saying it right? Yeah, endocardial action potential is longer. Here we have a stronger ITO, so the action potential is shorter, and here the upstroke is also faster. That's correct. Okay, so what do we see in HEY2 mice? What we see actually is that the epicardial action potential becomes similar to the endocardial action potential. And this could be because of remodeling or of flattening of ion channel gradients across the ventricular wall. We wanted to understand which currents were contributing to this effect. So we first looked at IK1 and steady state potassium currents. We did not identify any difference in those. Of course, one very good candidate is the transient outward current. So this is the situation of wild-type mice, where we have a larger ITO, transient outward current, in the epicardium compared to the endocardium. And this is what happens in the HEY2 heterozygous knockout mice, where ITO decreases in the epicardium, bringing it to be more similar to ITO in the endocardium. Of course, we also observed an increase in upstroke velocity. Now, ITO could contribute to upstroke velocity, but highly likely candidate for an enhanced upstroke velocity is a gain in sodium current. What we have here is sodium current in epicardium in red and endocardium in blue, again recapitulating what we ourselves had previously described in wild-type mice. And this is what happens in the heterozygous HA2 knockout mice. The epicardium INA is increased and reaches the level of the endocardium. So essentially, we think that perhaps HA2 could be involved in ion channel patterning across the wall of the endon part. And we believe that based on this data, it could contribute to Brugada syndrome, both through an effect on the sodium current and the effect on the transient outward current. And it's probably doing this by selectively impacting on the epicardial action potential. But now going back to genetics and the genetic architecture of Brugada syndrome and how can we find more genes. So we've been looking so far, the GWAS focused on the common variants. We're now busy with an extension of the GWAS. We're running a GWAS now in about 1,000 Brugada syndrome patients. However, what we intend to do next is actually go after the rare genetic variants. Will those play a role? An important question is, how many of these susceptibility variants can we identify? Don't forget, Brugada syndrome is a rare disease. At one point, we're going to reach the limit of statistical power. So that's going to be problematic, I think. And let's suppose we do identify multiple genetic variants. Will we be able to predict who is at risk of sudden cardiac death by means of a genetic risk score or something similar? What we're doing now is trying to push the boundary of genetic locus discovery to the whole genome. So if this is the genome, looking for rare variants, our search for rare variants has been in less than 2% of the genome. So our next work in the search of rare variants is going to be really looking into the whole genome by whole genome sequencing. So to summarize in some perspectives, we think that the genetic architecture of some of the disorders associated with sudden cardiac death may be more complex than previously appreciated. Genome-wide association studies, being for common variants and also for rare variants, may uncover novel loci that contribute to disease susceptibility, but also can provide us with leads to understand the disease mechanism. So as I said, so far we've been doing the common variants. We want to look for rare variants throughout the genome now. And I think we provide convincing data to show that HA2 is involved in patterning of action potential morphology across the ventricular wall, and it probably contributes to Brugada susceptibility to effects on both INA and ITO. And I would like to acknowledge my collaborators in this work. These are people from Amsterdam. Yuka and Julien did most of the work with the Brugada syndrome GWAS, Carol and Bremen and Oliver Kerk, Christian and Bas did the electrophysiology work. Also I acknowledge the group in Nantes with whom we did this Genome-wide association study in close collaboration. Manfred Gessler for providing us with the HA2 heterozygous knockout mice, and all the clinicians and geneticists in the different centers that helped us put together this cohort of Brugada syndrome patients that enables this work. Thank you very much.
Thank you. Questions or comments?
Thank you very much for this work. I really enjoyed the talk. My question is about one of your earlier slides with the South African founder and the QTC variability. If you look at those individuals, can you look at to see if any of them harbor the GWAS variants, and if that can explain some of the variability?
Yes, so actually I did not go into that today, but we've been doing similar work in the long QT syndrome. In the long QT syndrome, there's been a lot of GWAS published, and there are now, I think, around 60 independent loci that each have a small effect. So we're starting to look at them in aggregate. We've published already on the first 22 that have been identified, and we've looked at the role in modulating QT interval prolongation in a set of patients with long QT2. And we actually find that they predict quite nicely the extent of QT interval prolongation. So we use a genetic risk score as well. And with increasing genetic risk score, you see increase in QTc interval, having as much as 20 millisecond difference between patients in the highest quartile as opposed to the lowest quartile. We also tried looking for arrhythmia risk. We have a bit of a suggestive association, but by far not as strong as the link with the QT interval. So what we're doing now, we've set up another consortium on long QT syndrome patients, and we have also now around 1,500 programs with long QT syndrome. And we're genotyping those right now. We're genotyping everybody because we're very interested already in those 60 SNPs, and it's cheaper to do a GWAS than to type them individually, and we can then search all SNPs. And we'll see. I'm very curious. I hope we can predict arrhythmia, maybe in an algorithm, together with the extent of QT interval prolongation, age, sex, to try to build a composite score for risk.
Well, same question, but protective alleles?
Yes, so for every risk allele, the other will be protective allele. So you can look at it both ways.
Are any jumping out in the ones you looked at?
Sorry?
Are any things coming out of what you looked at so far? Even in the long QT?
Yeah, so this data we published on the 22 SNPs, and now we're waiting for the genotypic data to do Genomewide and to look at the 60 that in the JAM population were, yeah. But there are, absolutely, in the human population GWAS, it depends which allele you label. The other side is always then the protective, because it will be associated with shorter QT. Yeah.
So variations in SCN5A are associated with many arrhythmia syndromes.
Yes.
Are those exact SNPs associated with TBAC's binding inhibitor mutations, or are they different?
Yeah, so Vincent, I'm always telling him, overlay everything, and I think that they've found overlap between other signals and ChIP-seq peaks, but not for all, I think. Other arrhythmias, DSNPs, yeah. So,
for example, the SCN5A and SCN10A
Yes.
SNPs, are they the same that are AFib associated?
Yes, absolutely. I think 10A is by far the most interesting and the most robust for arrhythmia and the most reproducible. So we've linked it also to ventricular fibrillation in the setting of acute MI, and there are at least 3 studies linking it to AFib, always in the same direction, and funnily enough, in the VF and in the AFib, it is the conduction slowing allele which is protective, something which nobody understands, because intuitively you would expect, I think, that the conduction slowing would be the risk allele. But it's that SNP. It comes up everywhere.
So it seems so those, in the case of no gradient of ITO across the wall, it seems to be less of a phenotype in terms of ST segment elevation. Is that true?
This is what you would predict.
And that's my prediction. I don't know if you
Quite a bit. So loss of HA2 hastens conduction, but prolongs action potential duration. Based on the hastening of conduction, you expect that in the Brugada Syndrome patients, you have a gain of HA2 function, which will reduce conduction, but shorten the action potential duration then. So, yeah. So,
if you just think about the gradient,
yes.
And you have some natural experiments, like the guinea pig doesn't have ITO,
Yes.
so by definition doesn't have a gradient
Yes.
of ITO either.
Yes, and I think
It's interesting to see.
Yeah, and also, in the in vivo heart, how relevant is the gradient because of the
If you
coupling?
eliminate it in the mouse, it's anti-rhythm.
Yeah,
I wouldn't
Yeah.
think
It
it would
is.
be.
Yeah, no, it
yeah.
is.
So this is
The other question is, so there is a primary effect, we try to think about the primary effect. So you reduce expression levels
Yes.
of sodium channels. So or less of them make their way to the membrane. So,
Yeah.
but we know. So the immediate
Mhm.
assumption is it affects conduction
Yes.
through reduced excitability.
Yes.
But also, sodium channels co-localize in the intercalated disc with gap
Yeah.
junctions.
I think that's a
And the
very
question
good point.
is there any
Yeah.
secondary effect?
Yeah,
Because
absolutely.
we see fractionated
Absolutely.
electrograms and discontinuous conduction.
Yeah.
And maybe the fact that the architecture is not
Yeah.
different because of no sodium channels there
Yeah.
where they co-localize could
Yeah,
cause an
absolutely. It's becoming, I think, increasingly clear that if you disturb one element of the intercalated disc, be it connexin 43 or NAV 1.5 or desmoplakin or another desmosomal protein, that they really cross-talk. And I wouldn't be surprised if with decreased, well, I think the group in Utrecht actually showed it, that in the haplosufficient mouse, if I'm not mistaken, they have decreased connexins. Yeah, so that's another element. Yeah.
Any other thoughts? Please.
Do you have any data on early repolarization or anything else with J-wave?
No,
Okay.
no.
The small change that you see in the action potential amplitude, the obstructive
Yes.
velocity,
Yes.
sorry, it's a rather large change given how small the change in the
Exactly.
current. So
Yeah. Yeah.
I guess you
Yeah.
always think of the channels being there as
Yes.
sort of a safety factor than
Yes.
anymore. But is this suggesting that they're limiting? Is that the interpretation of why you're seeing those functional effects? It's sort of just surprising that it would be such a thing.
So basically, you're saying that the increase in action potential upstroke velocity is too much for the small increase in INA.
I'm just wondering.
Yeah,
I
you
guess-
know, the thing is this, I was actually surprised that you can actually find faster conduction. And we found it only in the RVOT, in a region which has a less conduction reserve, we think,
Yeah.
because of the reduced connexin 43 and its inherently lower NAV 1.5 expression. I think, yeah, I was surprised that we could actually measure faster conduction, but we measured it in an area where you have, I think, a lower conduction reserve than in other places. So maybe that is where it's limiting and where you could actually do with a little bit of a better push than an INA. Yeah.
Okay, I don't see any more hands, so thank you all.
Thank you very much.